So our first speaker is going to be Calvin Odero. He's with University of IFAS Everglades. He's a weed scientist, and he's going to give a little update on sweet corn weed management.
Okay, good morning. I'm Calvin Odero. I'm an extension weed specialist here at the EREC. And I'll give you some updates on sweet corn weed management. I'll just go over some of the common, commonly used herbicides that are out there. Research-wise, I don't have anything new that I'm presenting. I think I presented some information last year on use of pyroxasulfuron as a pre-emergence product. But today I'll just go over some of our commonly used herbicides here. So I'll just go over basic weed management in sweet corn, or whatever information like for this slide would be applicable to any other crop. So before what would constitute an effective weed management? The first critical important thing is correct weed ID. You want to know what you have out there in the field. So that will help you with selecting your proper control methods that you would want to use. And then based on that, you always want to use an integrated approach using multiple tools, not just relying on one tool. So typically around here, we do a lot of mechanical weed control cultivation. And then, of course, chemical control is a big thing with weed control management systems in sweet corn. And then we have cultural control, which involves rotating sweet corn with other crops. We do rotations with rice or sugarcane, or depending on certain other vegetables. So in terms of weeds, I've got a few weeds here that are commonly found in sweet corn. Can anyone take a guess of what these would be? That's why I put the big picture. Yeah, that's fall panicum. Typically, when it's small, it'll have a lot of hairs, like on the collar region. And when you open up the leaf, the ligule is going to be hairy. So this is, I would say, the number one grass weed that you'll find in sweet corn, plus any other crops that are grown in rotation with sweet corn, especially in sugarcane. But usually, as it grows bigger, the plant becomes much more smoother. And this one?
Anybody? We have these nice old mugs, if you figure it out. Come on. Anybody?
The stems are usually, the stems are flat. I don't know if you can see from the picture.
Good. No.
This is goosegrass.
Goosegrass.
Yeah, the stem is going to be very flat. And then if you open up the ligule will be membranous. So I would say the fall panicum and goosegrass are the ones that you typically find in the fields. There are other grasses too, but fall panicum would be the most problematic to control. And then for the grass or grass-like weeds, we have yellow and purple nutsedge. Can someone take a guess which one these would be? Because the easiest way to identify these, you don't want to wait for the inflorescence to be up. Usually you can look at the leaf, the tip of the leaf to tell the difference between the two. So which one would this one be? Yeah, purple, yeah.
Who got the purple?
Someone.
Come on, Joe.
Purple, and of course, the next one is yellow. So the purple, the tip of the leaf is, the tip of the leaf is pointed, but it's not like long. I mean, you get these one. For yellow, the tip of the leaf is pointed, but it's kind of a little bit longer. It's not like abrupt. So this sometimes is called like the tip is almost like a boat shape for the purple nutsedge, and then for the yellow nutsedge, it's slightly longer. So that's the easiest way to tell the difference between the two when they're small, when you're out in the field. And if you can't do that, then you can always try and pull the plants out. So there are those that have at the end of the rhizome, you find the tube attached at the end of the rhizome, and then the other one, when you find them, they'll be connected in chains. So which one would this one be? The one that is connected in chains. That would be easy to know. Purple, yeah, it even has the flower, yeah. So the purple would be connected in chains, and then the yellow sorry, the yellow, when you pull it out at the end of the rhizome, there will be one tuber. But these ones you pull out, then you find a chain, I mean, a group of plants. So that's the easiest way to tell the difference between the two. And for some of the common broadleaf weeds, these ones are relatively easier to know. You know, Spiny amaranth is a big problem here. There are other amaranth species. We have common lamb squatter, and then of course common ragweed. See a lot of common parsley, especially during the warmer periods. And I know people who, especially for some of our lettuce growers, common parsley is becoming very problematic, particularly for lettuce that is planted earlier in the season. So even for sweet corn that is planted earlier in the season with the warm weather, usually as long as night temperatures are higher than 65 and above, I mean, the plant just grows like crazy. And then we do have American black nightshade and ragweed parthenium that mostly you see it along field edges, but over time I've started seeing a lot of it, creeping into fields. So those, in brief, are some of the common weed species that you will find associated with the sweet corn crop in our area. So in terms of management, I know a lot of for pre-emergence herbicides, majority of them, to do a true pre, you really need to incorporate most of the herbicides. That's why I put the real rain here. Or if it rains, usually that helps. But if you plant sweet corn, like sweet corn that is planted later, I mean, let's say from February, March, when it's really dry, sometimes using some of these pre-emergence herbicides, really doesn't work, because when the ground is dry, there's no means to incorporate them if there's no rain or if you don't use irrigation. So I'll go through some of the herbicides that are commonly used pre-emergence. There are a number of them, but I'll just go through the list of what we would typically use around here. We have atrazine or Etrex, but mostly go by atrazine. And the rates are the use rates are different for mineral and mark soils. For mineral soils from 2 to 4 pines, and then for mark soils from 4 to 6 pines. And atrazine is mainly for annual broadleaf weeds, even though the label says that it can help with some grasses, but usually it's not as effective in grasses as some other herbicides. And it's usually ideal to apply it to moist soil after planting, so the ground has to be wet for the product to move in. But a good tank mix partner is usually dual magnum to help broaden your weed control. So if you tank mix atrazine and dual, that should take care of most of the broadleaf weeds and the grasses. And then Callisto is another one. I know most people use it post-emergence, but it's also labeled for pre-emergence use. The use rate for pre-emergence is higher than for post-emergence, so it ranges from 6 to 6.7 fluid ounces, and it's majorly good for broadleaf weed control. And it always require, at least some rainfall irrigation at least 7 days after application if you use it as a true pre. And Callisto can also be tank mixed with dual to help with the grass control portion of the weed control. And then dual, I know, is commonly used by a lot of people for grass control. It can have activity on some broadleaf weeds, usually some of the smaller seeded broadleaf weeds. And it also helps with suppression or even control of some of our sages. And usually if you use it as a pre, it's usually good to have some rainfall or irrigation to help incorporate it. And as I mentioned before, tank mix with either atrazine or Callisto can be good to help with the broadleaf weed control part when you're out in the field. So here is just a picture to show you how some of this product works. If this is an untreated field, this is where you use atrazine, and as you can see, mostly what is left is majorly grasses here, because most of the broadleaf weeds were controlled. And then the dual that is good for grasses, what you can see is mostly, this is common lamb squatters, and most of the fall panicum that you see here in the atrazine are controlled. And when you combine the two, you get a much better control, even though you get some escapes. So this was a field that was treated with this herbicide, but we had a very little rain after the application. So there's a big difference when you get good rain or good irrigation to incorporate the two products to end up with better efficacy. And then for our post-emergence herbicides, Atrazine still can also be used as a post. So in situations where the ground is too dry and you can use a true pre, I know mostly people use post-emergence applications, so the use rates are the same as when you use pre's, and it's going to be good for broadleaf weed control. As I say, the annual grass control is not as good, and it's ideal to apply them to weeds that are less than 1.5 inches tall. And tank mixes of atrazine with either Callisto, Laudis, or Amazon usually help, especially enhance control of your broadleaf weeds. And as I talk further, I'll mention the tank with Amazon usually is very good for helping with grass control. So all these herbicides are what are called bleachers. They cause bleaching in susceptible plants. So when you apply them out, you feel, they'll kill the weed. I mean, the weeds will turn white first, and then before they eventually die. So the next herbicide that is also typically used, you can also use Bazagran. Bazagran can be good for some broadleaf weed control, but it also can be used for sedge control. Typically, 2 applications are better to give you a better efficacy on sedge control. And Bazagran 2, which is in the same, it's what is called a photosystem 2 inhibitor, just like atrazine. It can also be tank mixed with Amazon, and that helps, especially enhance the activity of Amazon on grass control. So if you have a field that has a lot of sedges and grasses, a good tank mix would be one that has Amazon and something like Bazagran to take care of that. So that will take care of the broadleaves, sedges, and the grasses. And then Callisto again can be used for broadleaf weed control. It's a low use rate. Most of these the bleachers that I mentioned are very low use rate products. So you see Callisto is only 3 fluid ounces compared to atrazine that you're applying at 4 to 6 pints. And Callisto is very good for broadleaf weeds. It either need a crop oil concentrate or an NIS to help. Depending, sometimes you might see some slight bleaching on the corn, but rarely. And a good tank mix partner for it is atrazine or Etrex. And with Callisto, I mean, you can tank mix it with even up to as low as a pint of atrazine instead of using the 4 fluid ounces. Sorry, the 4 pints or 6 pints. So it helps reduce the amount of atrazine you can use on your crop, but still improves on annual broadleaf weed control. Then Amazon is the other product. It's also a bleacher. The use rate is from 3/4 of an ounce to 1 fluid ounce. It's very good for annual grass and broadleaf weed control. And it's usually good to mix it with either crop oil concentrate, mostly with an MSO, and it's usually good to put in some nitrogen fertilizer like a urea ammonium nitrate or AMS to enhance the activity. There might be some slight bleaching, but usually rarely. But a good tank mix with that would be atrazine too. And I'll also show you some pictures here to just illustrate how tank mixes of some of these what are called bleachers help and enhance the activity when mixed with the photosystem 2 inhibitors, either atrazine or the Bazagran. And then Laudis. Does anybody use this around here? Symphotrion. It's in the same group as Amazon or Callisto. And it's good. I mean, it's supposed to be for broadleaf weed and grass control, but I would say like Callisto, it's not as good as in grass control as Amazon. But you can also tank mix it with atrazine or let's say Bazagran to enhance its to give you enhanced activity. And Sandia, if you have sedges, is something that is used on sedge control in sweet corn or even in sugarcane or even in rice. And it might have a little bit, I mean, it has some activity on broadleaf weed control, but mostly it's for sedge control. So if you have a bad sedge problem in sweet corn, I mean, Sandia can be an option that you can use. And here are some pictures to illustrate some of those, the herbicides that I just talked about. So Callisto, as I mentioned, is really good for broadleaf weed control. Does not do much on grasses, as you can see here. You see all these grass escapes in the field. And when you mix it with atrazine, you can mix it with atrazine from even as low as 1 pint. Still does a very good job with broadleaf weeds. Here, they were a little bit, I don't know if you can see from the picture, there were some escapes of the broadleaf weed, but when you, with the tank mix with atrazine, all that is left is grasses here. And here, I mean, the control was even better with porpines. But usually, if you have a field that is predominantly broadleaf weeds, you can just use just a pint of atrazine with Callisto without having to go with, half a gallon of atrazine for post-emergence control. And then Amazon, as I mentioned, Amazon by itself, when applied at the right timing, I mean, you get very good broadleaf control. So this field had mostly lamb's quarter, some parslane, and then of course fall panicum. But you see, Amazon at announced really cleaned up most of the weeds. And then when you you can tank mix it with atrazine. So we had a tank mix with a pint of atrazine and 4 pints, and you can see, tank mixing with a high rate of atrazine really didn't make much difference. Just a tank mix with 1 pint was pretty good. So in a field where you have a lot of grasses, particularly fall panicum, if you apply this early enough, I mean, you'll end up with a very clean field with this product here. And I mentioned Amazon can also be tank mixed with Bazagran, particularly if you also have some sages. It really does help. You can tank mix with Bazagran. That will still take care of all your grasses, broadleaves, and still do some relatively good job with the sages. And Laudis, as I mentioned, it's labeled for grass and for broadleaf weed control, but just like Callisto, it's majorly just good on broadleaf. So if you look at this, you see all these escapes of fall panicum in the field. So those are some of the major herbicides that we can use in our sweet corn crop around here. There's really nothing out there that is coming up that I would say next couple of years. But I'm doing some work with what is called pyroxasulfuron, which is a pre-emergence herbicide that should be good on both broadleaves and grasses. But it usually does require some incorporation, just like S-metolachlor and the rest, to get some good efficacy with that, as when you use it as a true pre, especially for our max soils that sometimes get very dry. So when it's really dry, using a true pre-emergence, sometimes you're not gonna be doing yourself a favor, because you'll need incorporation. You'll have to either use some real rain to incorporate that into the soil. And of course, a lot of times, I know after getting that good level of weed control, we go back in and cultivate to do the healing and all that. So cultivation, in combination with the herbicide applications really also help. So I know most of you always like to have fields like this look nice with very minimum weed infestation when you combine either chemical or mechanical control. So that's the end of my talk. So if anybody has any questions, if some new products come up, I'll let you know. Hopefully something will come up. But the products that we have, main thing would be timing. It's critical to get good efficacy with some of the herbicides that we have in the market right now to get good control.
All right, so our next speaker is Dr. Mary Yong Cong. I hope I didn't massacre your name. And she's working with EVDAC DPIs, and she's going to talk about CAP sulfurida corn survey.
Okay, good morning, everyone. My name is Mary Yong Cong. I work for the Division Palang Industry Department of Agriculture in the state of Florida. I will have an update about our department in DPI. So this presentation, it was making for Crystal Ashman. He worked for in our group too, but in another region, and for me. Basically, okay, CAP's mission, CAP is a department who work with in collaboration with the federal agency. So we have budget from the state and from federal side. Basically, the surveillance, detection, and monitoring of exotic plant, that's our mission in all the state. And we have this organization here, another state have in there. So we survey all the targets. So we try to cover all the state. So we have three different divisions. The north side belongs to Crystal Ashman. The other person I mentioned before is region A, region B is central, and region C is south. That's my side. And there is another side on the west side of our state is over here. You see, like Palmetto, Tampa, there are two people working there, but both they are federal people. And we started in 2002 with only 2 positions for all the state. You can imagine how was the work at that moment. So in this moment, we have all the personal work for CAPS is on the table. So our side, our region, so Miami is 1 pest survey specialist and 1 OPS environmental specialist. She basically are doing just right now trapping for tomato commodity. So the rest is under my shoulder. So, okay, we do 3 different types of survey: trapping for insect and pests, visual survey for pests and pathogen. But basically, we're using different kind of trap, like for example, the yellow plastic and the Delta trap. And we use it to another sticky board when the crop start and finish. But basically, in all this day, we are using those 2 traps. And here in this table, I want to show you the scientific name and the common name and the type of survey we are making just right now. We do visual and trap. So as I mentioned before, the trap we use it, I show you on the picture before, and all these species are there we are looking for. So we using for all the different pests, we are using different lure. So that's basically we are doing just right now. And for instance, around Miami, we are trapping a lot just right now because there are a lot of nice crop in this moment. So one of the pests we are looking for in visual survey, we don't have trap yet designed for that, is the Abrotica speciosa. So the distribution is the Central and South America. We are, and there are 2 species basically here. Sometimes they are confusing. I think probably there is another, especially we will talk more about this. So because I know that identifier. So the major host is, as I show you right there, and one of them, of course, is corn. And the other visual survey we do is for Cyphamides, and the distribution is in basically Europe and Middle East, South Africa, and it was detected here in the United States, in California for the first time. And when I mention all of these pests, we don't have any. For instance, this year we don't have detention for any of them, so we are fine just right now. the host is grasses and cereal crops. And that's the additional issue is surrounding weeds and grasses and can use pests, and so they can detect it. Sometimes even when we place the trap, we can detect, the crops, they are really big and in different part of the tail. So basically I think the big crops, they are north, around the panhandle, and in the middle we don't have too much, and in south either. The other visual corn commodity survey is this species is a fungal pathogen, basically. And every time when we are going to the field, we try to make a work around the crop, and we are looking for different signs. And when we see something is different, we take the sample and send to Gainesville. All the sample, they are processed in Gainesville. So it's right there where we have the lab, the specialist, the identifier, and when the DPI, I mean the state, found one of the suspect pests, we send that sample to the federal side. So when we found something and we said, okay, we found X species, so it's because both sides, they identify this species. So that's the other one, visual survey about this species we are doing during this year and for the next year too. So every year we have a survey plan according to the intersection and different species we know they are around the country, according the interdiction in different airports, seaport. So we receive every month all the intersection, and after that we have an idea what's going on around us. So another, this starting with trapping just right now, Autographa gamma. We use that kind of trap, the plastic, yellow plastic trap. And inside we put a killer strip, just in case when some of the moth or butterfly insect, they go inside, are killed. When we take it out, so we are safe to move this sample. And then on the top, we put the lure. And in this case, we use a Autographa gamma lure to try to find. So we didn't find nothing in this about this species. It was we found many intersections, but nothing on the field. So we are good. The other one, this is another kind of trap we are using. So, the name is Delta trap. And inside the Delta trap, we put on a sticky trap, and we glue in the sticky trap the lure. So that's trap work very well. So is to try to find Taumatotibia leucotreta. And we have, every time when we found a crop, we try to put it for different targets. So if I found, for instance, one crop, I try to place, consider the distance and the size of the crop, I try to place different trap for different targets. So it's not only one trap. And the host over the 50 species, so you can see there. So we have this, I show you just right now about corn, but we have for instance, this species we place trap for. We have one of the survey we have is about nursery, so we place it. For instance, sometimes the same trap and the same lure for another host. And this is another. Okay, that's the sign about the sign. Yeah, let me. Okay, this is another species. So we use sometimes this sticky yellow trap, and when we place it, that's a sticky board, and we use it, for instance, at the beginning and at the end of the crop. So we need to follow how it's going on, everything. So that's we need to know when the crop started and when it's close to the end. So it's native from Europe, Africa, Asia, and Oceania. And right here, that's I present here some of the data we have for this year. That it was until I think in November. Yeah, we process all the sample. So we have the visual and about the another species we are trapping. So that's the number. And we continue doing that during December. For instance, I remember I sent like about 10 different samples to Gainesville. So they are not included right here in this report, but every week the people are submitting samples to Gainesville. The people working north, central, and south Florida. And that's it. So that's my contact information. If sometimes you need to submit sample, Anna have my contact information. You can send the sample directly to Gainesville, but the best way is send to me and I submit the sample to Gainesville because I know very well where that sample have to go. So you contact Anna, or this is my contact information, my email, and can submit the sample directed to Gainesville. Okay, thank you.
Thank you, Mary. Anybody has any question? And then, like Mary said, you can find information online to just type FDAX sample submission, and you can use that form. But like she said, the simplest way, probably contact me or her, and then she's going to send the samples to Gainesville.
Yeah, there is.
They are doing all the identification.
Yeah, sorry. There is a specimen submission form online in DPI webpage. But the most important, is you send me the location and the GPS coordinate. You can take it with your smartphone, and the collector and the date. That's the most important information I need to submit a sample. Okay, thank you.
Thank you again.
Yeah.
Yeah, so our next speaker is Katia Xavier. She is an assistant professor here in plant pathology at the station. And then she is going to talk about sweet corn diseases and management update. Thank you, Katia.
Hi, can you all hear me? Good. Good morning. Okay. Thank you, Anna, for having me here one more time. And I'd like to thank you all for being here today and trying to learn a little bit more about sweet corn, right? Different strategies, like how to control weeds, how to control pathogens, and all of that good stuff. So for my talk today, actually, I'm basing on a presentation that Dr. Reid gave last year. So some of the diseases that were important by that time are still important, and I'll be talking about them. and also about the management strategies, especially about the chemical control. So that's all based on Dr. Raid's presentation, and I'm going to point that out when I get to that part. And also I'm basing this presentation on a survey that I did in April of this year. So during the spring, I started this job in February, and when I got here, I saw a lot of northern corn leaf blight in the field, and I'm starting to understand it a little bit more when this disease appears. Some are more present during the fall, some during the spring, and it depends on the weather and several other aspects. And anyway, I did this survey, and the results, excuse me, were that the most, oh, sorry, the pointer is here. Is it working?
Yeah,
It's working. There is a little delay, like 1 minute later it shows there. Anyway, so the most important disease that based on this survey is northern corn leaf blight. And also you can see here on this picture that Matt Burden from Glades Crop Care took last year. He sent me that picture, and I'll talk a little bit more about that later on the presentation. But another important disease, the second one is southern corn leaf blight, and then common rust and southern rust. And I'll be talking more about these diseases. And actually, I divided this presentation based on those diseases. And also, Dr. Raid last year talked about the northern corn leaf spot. And I added the tarry spot, that's a disease that I'm working with now. And I'll explain a little bit more about that disease in the end of this presentation. And also, in the end, I'll talk about some of the management strategies that are recommended for those diseases that I divided in 3 groups. First blights, and then rusts, and then spots. So for the blights, and I put this disease cycle here because I want to illustrate how this pathogen can survive, and it actually survive on the crop debris as a mycelia or conidia. And then in the beginning of the season, those spores can be released here, and they can be disseminated and spread through the wind and also rain. That helps that. And then it's going to land on a leaf surface, it's going to start the infection. That pathogen is going to sporulate, produce more spores. And then from that first infected plant, it can move to other plants in the same field, and it also can move to another fields. So southern corn leaf blight is caused by this pathogen called Bipolaris maides. And it's actually considered to be a disease of warm to hot temperature. So it's favored by temperatures between 20 to 32 degrees Celsius, you can see here. And this disease, we can see it during the fall and also in late spring, and it makes sense because it's when it's hotter, right? And also this disease is aggressive on both new expanded leaves and also older leaves. So it kind of differentiates it from the northern corn leaf blight. And the symptoms also are totally different. So if you see those pictures, those are tiny little lesions on the leaf surface. And it actually, there's another disease that's the northern corn leaf spot that's similar to this one. Excuse me. And the pathogen actually that causes this disease and the northern corn leaf spot is the same genus. So if I look under the microscope, they just look the same to me. So I think who is more experienced and have seen this disease more in the field, they can actually see the differences based on the symptoms, but I'm not quite there yet. Probably Dr. Raid could do it with his eyes closed, right? So I'm learning. So as you see those diseases and you have doubts, you can bring samples to me and I'm going to analyze it. And also maybe in a month or 2, we will be able to do some molecular work. So that's going to help to detect these pathogens, and then I will be able to actually differentiate them better based on some molecular tools. Okay, and for this disease, in terms of resistant varieties, it's available, but it's not very common. And now the northern corn leaf blight. So that's the one that I showed that picture in the beginning. So you can see how the pathogen infects, especially the older leaves. It's a blight, or it seems that they just burn the leaves. It's incredible how aggressive this pathogen is and how it can kill the plants. And I saw it with my eyes during the spring, and I was like, wow, this is quite something, you know. Anyway, it's a cool to moderate temperature disease that's favored by 18 to, I think that's actually 28 degrees Celsius. I missed one number here, but, and it's most commonly observed during the spring, as I said, and on the older leaves. Now I'm going to talk a little bit about the rusts, and I actually put the disease cycle here again, because this pathogen is interesting because it needs the host to be alive. So the pathogen also can be alive and can multiply and then produce more spores and infect other plants. Now, if you think about the summer, we don't see corn plants here, right? So you think, okay, if we don't have corn plants, this pathogen cannot survive. So where is it's coming from when we start the season again? And it's interesting because this fungus can actually overwinter on some tropical and tropical regions on this weed here. And then from that, it can be disseminated through the wind, let's say Central America. It can come here and it can actually land on a plant surface, and it can start that disease cycle in here. So one spore lands there, it causes disease, it sporulates, and then it can move to other plants in that field and also to other fields. So, and then it can just spread and be disseminated by the wind. And then in the end of the season, it does not survive on crop debris. So that's actually one of the strategies that's recommended for this disease. If you have a cornfield here and want to plant another one here, just be careful because that disease can move through the wind and the blights as well. And the common rust is caused by this pathogen that's called Fuchsinia sorgi. And it's a cool temperature disease, which is favored by temperatures between 16 and 25 degrees Celsius. And this one is most common during the spring season. Like we have some cornfields here at the station. I was looking for those rusts. I couldn't find common rust right now. What I could see is southern rust. So how can we differentiate between those two rusts? So if you look at this picture here, oops, here, you see that's more like a cinnamon color, right? It's more like dark brown color. And I'll be showing the next slide, the other rust, so you can see the difference between those two. And also another difference is that this one is most aggressive on those new leaves, and the southern rust is more common, like it's common in both new leaves and old leaves. So you can separate them based on that. So now just take a look at this picture, and I'll put the next slide, and you can see the difference in color. See, this one is more like an orange color, more like a rust, right? That we think about. So, and this one is more like a warm to hot temperature disease, which is favored by temperatures between 23 and 33 degrees Celsius. So it's mostly common during the fall, as we can see it now on the plants here, and also late spring. And this one, as I said before, it's aggressive on new and also old leaves. So if you're scouting for the disease and you see on new and old leaves, that's another sign that's southern rust and not common rust. Now about the spots, this is the northern corn leaf spot. As I said before, the pathogen that causes this disease is the same genus, a different species, but same genus as the one that causes the southern corn leaf blight. So for the identification, as I said before, it's a little bit complicated because the spores just look the same. And this disease is more like a cool to moderate temperature disease. And it's favored by those temperatures between 18 to 27 degrees. And This one appears more during that midwinter. That's more like the transition between fall and spring. And this is aggressive on young tissue, young leaves. And those are, oops. And here are the symptoms of those, of this disease. Those lesions, they are small, very similar to me at least, is very similar to the southern corn leaf blight. Now, tar spot, it's an emergent disease in the United States. And to me, this disease is really interesting because it was first reported in Mexico in 1904. And then it was in Mexico, all happy there for 100 years, and never got here, right? And then suddenly they reported the disease in Illinois and Florida between 2015, 2016, and boom, it just exploded. In 7 years, the disease was already present in 14 different states. And if you look, and that's on field corn, okay? But if you look here, this map, the gray areas, those are all the counties where they reported the disease in 2022. So it's incredible how much this disease has spread within the United States in just 7 years. Those are the symptoms of the tar spot on field corn. And I saw this disease here also on sweet corn this year, but was not very aggressive. So it's way more aggressive on field corn. And on sweet corn, it's just show a few spots, but it's not that aggressive. Anyway, we have a project. I have a project together with Dr. Marcio Resende that's actually to identify some solutions before this disease becomes an epidemic in sweet corn in the region. So what we want to do is to screen some sorghum hybrids or varieties or germplasm, looking for some sources of resistance against this disease, and also screening for fungicides to see what can be used in case the disease becomes a problem in a near future, right? Sorry? So it spreads through the wind. Yeah, but they have also identified that this pathogen can survive on crop debris, even though it's an obligate parasite like rust, or they need the alive host to multiply, right? But this one also can survive on crop debris, which was a surprise for them, for the researchers that did this work, because they don't know exactly how the disease got here in the United States, right? Was it wind? Why it was in Mexico for 100 years and never got here? So people believe it's wind, but also there are some people that believe it came with the seeds or something like that. In terms of management strategies for the sweet corn diseases, I'll be talking about chemical control, cultural practices, and host resistance. And for the chemical control, as I said before, I'm basing on Dr. Reid's recommendation. So what he recommended last year, and I believe it's still the same, is to apply protectant fungicides and also some systemic fungicides. So in terms of protectant fungicides, he recommends sulfur, mancozeb, and chlorothalonil. You just have to pay attention and read the label because they have different pre-harvesting interval. So by reading the label, you can be sure that you spray it in a certain time that is not going to have any residual when you harvest that corn. In terms of systemic fungicides, the triazoles are recommended for the foliar blights that we talked about. And the estroberulins are recommended for both rusts, blights, and also spots. I didn't put here. And the carboxamides, what Dr. Reid was recommending here was actually to do a pre-mixtures. And he has this table that I'm not going to go through, but if you want this presentation later, you can have it. Anna can make that available for you. But in this table, he has the combinations that he believes are the best, you know, because if you mix different chemistries, probably you're going to have a better control, especially if you spray one time and doesn't control the disease, maybe you want to another active or another chemistry. So by mixing them, you can have a better control of those rust, blights, and also spots. So that's it about the chemical control. Now for cultural practices, it's important to stay clean, start clean by selecting a field that has no corn debris, because those blights can survive there and can be a really big problem. If you have a field in one area and you have a problem with one disease and you want to plant corn there again, just be aware of the disease you had before, because most likely that's going to be a problem, you know, for the next field corn, for the next corn you plant in there. And be aware of surrounding areas. If you have a young field here and another one that you're harvesting there, and you have lots of disease there, just be aware, because that can move through the wind to the new field. And it's really important to scout early for those diseases and throughout the season. And with that, just try to stay ahead of those diseases, because once they are there and once they become an epidemic, it's really hard, right? They are going to have some big losses. So stay ahead of the disease. And we are here to help. You know, if I don't know, I'm going to study, I'm going to look for information, and you can contact Anna as well, so we can work together trying to identify solutions once you find a disease in your field. In terms of host resistance, there are some resistant varieties against common rust, northern corn leaf blight, and also northern corn leaf spot. So that's actually another option. And with that, thank you for your attention. And if you have any questions.
Hi there. Can you hear me?
Yes.
You mentioned screening fungicides for you know, tar spot control. Is any work going to continue on the leaf blights as well, or in tandem with that?
Yeah, so the idea here to screen fungicides would be to actually study some that the growers are already applying against northern corn leaf blight, southern corn leaf blight, and northern corn leaf spot, to see if we could use the same ones to control tar spot, if they are going to be effective against tar spot as well. Did that
Right,
answer
so
your
we're
question?
expecting we're controlling tar spot and sweet corn going after the leaf blights, but that's where all the, you know, where all our attention is on the leaf blights. And tar spot might be a, you know, like a global or a nationwide concern where, you know, very little fungicides being applied, but in sweet corn, yeah.
In
it's the
sweet
blights.
corn, yeah, usually the growers spray against other disease, right? So most likely it's going to control tar spot. And maybe that's why it's not a problem on sweet corn yet.
Right.
Because, you know, it has been sprayed with the fungicides already.
Right, but continuing efficacy research on the blights is, you know, our kind of top concern.
Yes, I think I didn't mention, but another project that I want to work on, and we are actually working on that project, like writing it now, is to study, like that picture you sent to me, right? You said that the growers tried to spray and did not control the northern corn leaf blight. So one of the goals we have is to understand why those chemistries are not working against. And actually my first slide, let me just show that really quick. I forgot to say that, but it wasn't my first slide. Here. Okay, anyway, so if the fungicides are not working, we want to understand why. Did the pathogen population change and now have some mutations on the site where the fungicide should work, and that's why it's not killing those pathogens? Or maybe the pathogen overall create a new race, and maybe that's why the fungicide is not working. So that's another project that we have that we are going to do on northern corn leaf blight. So in fields like this that you send me some samples, what I did was I stored those isolates. And once I have the funding available, what I'm going to do is to screen those isolates and see if, like in the lab, did that fungicide still was supposed to be effective, or did it work in the lab? So it should have worked in the field. Is it a new race of the pathogen? We can do those studies once we have those isolates. So every time you send me samples, for those disease, other crops, I try to save the isolates if I can grow them in culture, so we can have this kind of projects later. Did that answer your question?
Yes, thank you.
Okay, yeah.
Any other questions? Thank you, Matt. Anybody? No. All right, so thank you, Katia. So here we go again. So this is the basically the verbiage that we receive. We need your input, and it's true. We need your input because we cannot do it by yourself. These researchers, we are basically looking looking for your help to share information with us about impacts experienced at your farm and ranch and other operations. So when you click on these surveys, you will see they ask for your consent and they ask for all the details, and it's really important to us, and not just for us, but for you. And so what's the goal? Why we are using this? It's because it's used to rapidly assess agricultural damages and losses to inform industry associations and local, state, federal government representatives as they make decisions related to formal disaster declarations. So that's why it's important, because you can, if you report these things, it can make things easier, and then this decision can be made based on this. So disaster reliefs, response and reliefs can be done. And so also what's happening when we receive all these data. So all these data information is received and summarized and provided to leadership within the state level industry associations, to FDACs, to USDA offices, and FEMA. So basically it's used for quickly communicate the size of the scope of overall losses. So they try to just figure out what's going on, because everybody wants numbers, everyone wants data, everybody wants to know what's going on after a hurricane or after a freeze event. And if they have enough data, they can even summarize everything by county and community group. And the good news is, because this survey study is approved through the Institutional Review Board, we call it IRB. So if it's approved by IRB, it's protected as a research. So the individual records do not have to be released, even if IFAS receives a public record request. So you are protected. So IFAS asked help from FDACs and Farm Bureau, and they are distributing these surveys to. We are helping them via email, Facebook, Twitter. So if you saw something like this, don't be afraid. This is something that this data is going to be used for you later. So it can the state can use the data to give you a relief package or a relief fund. Sorry. The only bad thing is about it that producers still need to report everything to their insurance. So it's kind of double reporting, but used differently. But the main thing is that even for Michael and Irma, these data was used to make federal level decisions on Florida qualification for the amount of disease relief that basically we received. So what came out actually after Ian, if you see this, that considering all the information that they processed, they believe that the losses for agricultural producers, crops, livestock, animal product, it resulted between $787 million and $1.56 billion. It's a wide range. You see why it's wide range, because we have no idea, they have no idea. It's hard to estimate. How you can estimate something when you have to stop planting for 2 weeks, how it's going to count at the end of the day when you look at your yield. So about Palm Beach County, when a hurricane is coming in the Atlantic, Matt Van Wilden sends me an email, hey, we need to get together because we need to report on this. So how we do it, I cannot do it by myself. So I rely on you, I rely on crop consultants, I rely on growers. I am communicating with Tori, with FFVA. We try to work together. We try to figure out the way how it can be more efficient, because I cannot drive around all Palm Beach County and figure out and do site visits. So whatever you guys are doing when hurricane comes and then you see a text message from Anna, oh my, yeah, again, I have to do it. I try to protect the growers. I try to protect the crop consultants too. I don't put name to my reports. I don't put exact numbers to my reports, but I try to kind of summarize what's going on out there. So
So when you see this, because you see these things, and I try to, it's not nice. And but among all these, at the end of the day, we try to work together and then figure out a way how we can report something but not hurt anybody with it, because this is important. And at the end of the day, if you look at what FFVA puts out, it's kind of vague too. It's always on a positive note. You know, growers will continue working hard to ensure Florida will be ready for the fall harvest season. They got hit hard. Some of us got harder than others. If you look at what happened, Ian, on the West Coast, you cannot compare it to the East Coast. So it's always site specific. So even when I'm doing my little report, I try to break down Palm Beach County, because we have a team leading Ron Rice and Matt Van Wilden, who is the sugarcane and rice extension agent, Frank Dowell and John Roberts, who is the environmental court agent. His job may be a little bit easier. He's working with the nurseries. I'm working with the fresh vegetable producers, which is a little bit always more sensitive. But, you know, I try to put this little report together so even the county can be aware what's going on, even if it's not precise, even if it's not perfect. But, you know, I have a little summary for the EA. I have a little summary for the middle part of the county, and also for the ag reserve area. So just I wanted to thank you all the crop consultants that can help me to summarize these things. And we try to find a way with Tori how we can make this a little bit more efficient. So we don't bother you like from 3 different directions as growers that we need information. Can you call us back? So we are working on it. And so thank you for all your support and all your patience. And Tori will talk about a little bit why it's important to give good information and not false information, and then also how we can probably make things better. So I will just give this to her. And if you have any questions, I mean, please let me know, and then we are here to work together. Let's see. Here you go. So yeah, Tori is the commodity service and supply chain manager, and then she's always also worked with the FFVA.
See if I can make this work. All right, so I'm coming in with the assumption that everybody knows that what FFVA is. Is that a pretty good assumption? I see some nodding. So real quickly, Florida Fruit and Vegetable Association, we are a full service specialty crop organization. We serve every specialty crop, sugarcane and sod included. And we have basically, I like to call ourselves experts. I don't know that that's the right word, but on labor, pesticide registration, we've got water and natural resource management. So if growers need anything, they know that they can come to us. So that's kind of where I'm coming from as I start my presentation this morning. So I'm going to build on what Anna just talked about, and then just a couple other things that FFVA is watching that I know is also on y'all's minds. So first, again, going to talk about disaster response activities and the purpose, and then some current issues that I know I'm watching and that our leadership is watching. So Anna just talked about the big events this year were the January 2022 freeze, Hurricane Ian, and Hurricane Nicole. Obviously, other things going on. But you can see from these headlines that what happens to Florida crops does not just stay in Florida. These headlines go nationwide. They sometimes, depending on what crop it is, will go worldwide. And so these headlines sometimes get out ahead of FFVA, and they start kind of speculating on what's going on. And so our job is to basically say, yes, and here's what's the actual situation on the ground. So you'll see the Packer, AP News, and Capitalist are up here, but we've got CNN, Fox, ABC, NBC. They're all coming to our communications team to figure out what's going on in the field. So this is really why it's that accurate reporting and really the accurate messaging becomes even more important, especially when it comes to market impacts. And I'll talk about that in just a second. So Anna and I work very closely on assessments. Craig and I also work together on assessments. And one of the large conversations we're having right now is how do we do that without burdening the grower or burdening more extension agents or researchers or whoever that is. So when we talk about storm assessments from an FFVA perspective, and again, mine doesn't have to be as formal as Anna's does. Mine's a lot more kind of narrative, and I can just kind of get off the record with the growers, and they know they can tell me things that are not going to get reported, but I can kind of doctor them a little bit to make sure that they're a little more anonymized. So when we talk about pre-storm planning, we're determining immediate needs and we're planning for future outreach. So these are the questions that, you know, everyone is asking, not just FFVA. So if Tori from FFVA calls you and says, what's going on? It's not just me, it's, you know, CNN's already asked these questions, so we're coming to you with them. So how big is the storm? What type of storm? What region? Wind, rain, or both? And that became really important in Ian, especially as we started doing program planning and how do we help growers? You know, maybe wind speed didn't hit your farm, but you had a ton of rain. And so how do we make sure that we tell that story? What crops are in the path? What part of the season are we in? And then when is the appropriate time to start reaching out? So in a pre-planning, that we want to make sure if we're talking to growers or crop advisors, we know y'all are in the field. If you're an input supplier, we know that you're trying to make sure you're in contact with the growers to get them what they need. So when are we going to actually start reaching out? The other piece of this being, as we go into the post-planning, we know that weather events are going to take days and sometimes weeks to actually show the damage or recovery. So we're kind of thinking about that too. So during the storm, we try and leave you guys alone. Post-storm, again, that's when we start thinking about, okay, what's next for what our growers specifically need. So the first thing we always ask is, are our members safe? We don't care, you know, about the crop assessment before we know that the growers are safe. And a huge part of that too is, do they even have power or internet to get in touch with us? If they don't, we're reaching out to Extension or other people to try and figure out how to get in touch with them. Then we start talking about when should we reach out, what information is already available is a big piece of this. If we know that there's reports coming in and I don't have to reach out to, I'll pick on Cooper. I don't have to call Cooper right now because I kind of know what's going on. We try and hold that back for the first 2 or 3 days so that we're not, you know, inundating people with calls. And then what do we need to know is a big one too. Sometimes that question is we specifically need to know what is the % loss? And I will call Craig and say, Craig, what is the % loss? But sometimes it's just, you know, what's the market implication? What does the market look like for you right now? Was there a crop loss? And if there wasn't, is there another issue? Is there a transportation issue? Or is there an input issue that's going to be in the way of you getting back on your feet or replanting? So the type of assessments, I thought this was important to include. Anna just talked about these crop loss assessments. So this is what her team is doing for the most part. She's going to be working with Dr. Krista Court's office. She's going to be talking specifically about percentage crop losses. They dig into those numbers on the actual dollar losses. Narrative assessments is where I think we kind of fall. And so we're talking about overall effects. We're pressure testing those compiled dollar losses. So a lot of times what will happen is FDAX or UF will come to FFA after they have those numbers and say, how does this feel? Does this feel like a good number? And then the information is going out to media, lawmakers, FDAX, grower members, and trade members. So what is the data used for? Anna talked about this a little bit, but all that data is anonymous when it comes into us. But the biggest thing that it's used for really is storm recovery programs. So as soon as a storm's coming through and even beforehand, our staff is looking at what kind of program is going to help growers either replant or with infrastructure losses. When you think about Irma, there was the citrus block grant that helped a lot of the citrus growers. So how do we build those programs to help with the need? If you think about even CFAP, during the pandemic, we were going out and granted it wasn't a natural disaster, but we were asking what were those impacts and how can we help? Improving crop insurance products is one I hadn't really thought about really until, you know, Ian kind of came through. I had never heard of this Hurricane Insurance Protection Wind Index. And all of a sudden it was the most important crop insurance program out there. But then you start talking to growers that say, you know, I had this crop insurance product, but it didn't help me because X, Y, and Z. So now we're taking all of those X, Y, and Z's and going back to risk management agency and saying, yes, you had a crop insurance program, but it didn't help because of these issues. And so that's where that information is also helpful. Media outreach, we already talked about that. People read those stories a lot more than you think they do. So just for example, my mom's a dental hygienist and people kind of know what I do. I don't think my mom even knows what I do, but people will come to her and say, what is Tori saying about tomatoes? And there are people who have no idea what's going on, but they're reading the news. So that's why these are also important. Legislative outreach, I always point this one out because if you've ever met Mike Joyner, who is our president, he's best friends with everybody, but he'll have the governor, he'll have the commissioner all calling him and saying, Mike, what's the damage? What do we need to do? And they want to know basically 6 hours after the storm has moved out. And so a lot of times we have to tell them we don't know yet, but we know that a program is going to help and we will help build that. So when we reach out to you guys, that's what we're looking for, is what we can actually do to get money that will be helpful. Future disaster declarations, again, some of those gaps that you guys are seeing when a storm comes through, that helps us with these future disasters. And that includes like weight limit waivers and things we might not have thought of for this storm, but we should think of for the next one. Biggest thing here is we never want to swing the market by overstating damage. Frank, I'm looking at you. So not that you've ever done that, but one thing that we talk about internally a lot is, yes, we know that there's damage, but what we don't ever want to do, and Anna talked about this with the release that was up from FFEA, we never want to say total loss, no, fruit and veg from Florida, it's all gone, until we actually know that. If it's the case, then we want to tell that story. But if it's not, and we think some of the crop might come back, we want to be very careful about how we word that, because y'all all know if the buyers are gone, they may not come back, and we never want to be in that situation. So this is just a question I would pose. I'm not going to take questions right now. I know we're kind of on a timeline, but we're working on how we can better support the industry during storms. So Craig, Anna, and I have already started these conversations, but we know that getting calls from 5 and 6 people after a storm and telling the same story is not very helpful to you. You don't have the time to be taking those calls. So we're trying to figure out a better way to kind of to gather that information. And I'm really excited about what Dr. Krista Court's doing up at IFAS. I think that she's got a really good idea on how to, look at wind impacts, but she's also looking at water impacts as separate, different impacts. And I think that's really helpful for future storms. So keep that in mind. I'm here all day and I would love to hear more about how you guys think we should help. Last thing I want to talk about, and I have no idea how I'm doing on time. Okay. It's just an FFEA current issues update. There are so many issues that FFEA is watching right now, but in my role in the supply chain and then also in the kind of commodity services world, trade and supply chain are the two that we're talking about every single day. So kind of our tagline on trade is that we need immediate, effective, and enforceable solutions to restore market fairness for U.S. producers of fresh fruits and vegetables. I'm assuming, again, that everybody kind of knows what we're talking about here, but in the trade world for fruit and veg, we talk a lot about influxes of imports from Mexico that are coming in, especially in our spring seasons on top of several crops in the state. So that's one thing that we're watching closely right now and trying to get some help. And then on supply chain, ensuring a reliable labor force and prioritizing the easing of grower input costs. So on trade, again, what are those enforceable solutions? This is a top priority. It's more important than it ever has been before, as we're hearing growers say, I'm out, I can't compete. And then they look to us and say, what can you do? So we have the USTR panel on seasonal and perishable that came out of our Section 301 petition. We're working with USTR to build this panel. The charge of that panel is basically going to be to look at the seasonal and perishable product issues in the Southeast and try and come up with a solution for us. Risk mitigation efforts, we're kind of looking at how do we come up with either a crop insurance type of program or some type of payment program to help growers when their market prices fall below maybe a historical level or even a production cost level because of imports. And then USDA data integrity. So when we look at crop insurance and, if we're going to talk about crop insurance as a potential solution or even to these disasters, the data that USDA uses to build those programs becomes even more important. And what we've heard from growers and from several of y'all in the room is that a lot of the data, especially that AMS has, might not be the best reported data. So how do we improve that so we have better products? On supply chain, these are just 3 of probably 40 issues we're hearing. And I have a meeting about this tomorrow, so I'm hoping to be able to update some of this and have some action items. On inflation and input costs, we're hearing 30 to 40% input cost increases. Depending on who you're talking to, that number goes even higher. And growers aren't receiving that back in their sales prices. And that's been told loud and clear to us by our board of directors. I, would like to have a solution to that, but we're still, again, tomorrow going to talk to some of our growers and say, how do we make sure that you're getting what you should get to make sure that you stay in business? We're watching consumer buying habits changing after the pandemic and with inflation. And knowing that all of these issues are global issues that don't have simple solutions. Look at transportation issues. Somehow the driver shortage is all we talked about for the last, 10, 15 years. This year, for the first time, diesel costs have overtaken the driver shortage as transportation industry concerns. That's scary to me. For that reason, we've got shipper prices that are actually returning to, more 5-year averages. So you might be seeing, a little more average shipping costs, but their carrier costs, because of those diesel price increases, are approaching the cost to transport. So what that means is you're seeing some of those smaller carriers maybe pull out because it's not as profitable for them. So we're watching kind of the impact from that. And then just changes in grower planning. This is no surprise, impossible to budget ahead. Gone are the days of next day ordering on inputs. And I know there's a lot of input suppliers in here. And so we're just encouraging our members to communicate early and often with their suppliers. So these are just top of mind, and we're working on these issues tomorrow. Hopefully I'll have some solutions. And this is my information. Please shoot me an email if you ever have any questions, member or not. I would love to get to know you. And I have a lot of our emerging leader graduates in here, and that's really exciting to us to have you all together, and makes me feel more comfortable. So thank you guys.
Thank you, Tori. Anybody has any one quick question? Yes, we are one. And she is going to be around, so feel free to reach out to her or me. I have this little, basically that was a preliminary assessment report that IFAS put out. If you want to have a copy, it's available online, but you can take this one too. And then this is probably that was our little report from the county on Hurricane Ian that Matt Van Milden coordinated, and then reach out to him too, because he was coordinating this little team to put this report out, and then he's working with us with, like I said, John Roberts and then Frank Dow and myself to put this report together. So anyway, thank you again, and then let's move on if that's it. I have a pre-recorded presentation from Marcio Resende, who is the sweet corn breeder up in Gainesville, and he's gonna just give a little update on this. Let me see if I can launch this.
Anna asked me to give you some updates about the breeding program during 2022. I'll just share some brief updates. We are in the process of wrapping up the Sweet Cap grant. I talked about this project before in previous years, and this is a project that was funded by USDA and initiated in 2018, and included 5 different institutions: University of Florida, USDA, University of Wisconsin, Washington State University, and Iowa State University. So the project will finish in the next few months, and we're in the process of writing final reports and so forth. There were a series of outcomes. I think this was a great opportunity to bring together a lot of different researchers from different areas, from different locations of the country. And we were able to screen and identify sources of good traits for sweet corn breeding, both southern cornleaf blight, northern cornleaf blight, virus resistance, ear traits, tip fill, and seed germination under cold and regular conditions. So it was really one of the large outcomes was screening for a diversity of important traits, screening materials that could be good donors of this trait in the future. And the project trained several students, and we're now in the final stages of it. I reported to you one of the tools that we were developing in the context of this project. It has now been published, and it's really been used operationally in the breeding program. And that's what we're calling ERCV. So that was a software that was developed in my lab. And now every ear that's produced in the context of the breeding program, we snap a picture of it, and the software will use artificial intelligence to identify where the ears are in the picture and measure them. So we have a very objective and more precise measurement of several traits, such as the number of fancy USDA fancy ears, and then how much tip fill you see, what's the exact curvature, which is a trait we obviously don't want. And we actually can now quantify all of these and remove them from the program, traits that are not of interest. The good news for 2022 is that USDA agreed to fund the second round of SweetCap, which we're calling it SweetCap 2. So this is a USDA NEPA funded award, $7.5 million for the next 4 years. And we now included a few new institutions. So the project now has 7 participating institutions. I am the project director, and UF is leading this initiative, but it also includes researchers from USDA, University of Wisconsin, Washington State University, Iowa State University, Clemson, and Michigan State University. The objectives of this project are pretty broad. They include targets of interest not only in fresh market, but also in processing sweet corn market, not only in Florida, but also in the other production areas, such as the Columbia Basin. And this group of researchers, you recognize here, Julien will help us and will carry research in insect tolerance and insect behavior. We have now Wendy Daw joining the program, the project, and she's a nutritionist and has a lot of research experience. Survey the consumers for their perception about food. So in this context, what we want to understand is how consumers view sweet corn and their preferences, and whether they're perceived as a healthy food or not, and really understand consumer behavior for sweet corn, and how the millennials and the new generation is looking at sweet corn as a vegetable for consumption. So just as a brief summary, so the project has 5 objectives, and one objective number 1 is to develop breeding technologies. So this is identifying ways by which breeding, which is a long process, can either be improved or accelerate. So how can we get to better hybrids faster? And that's a lot of what this objective 1 is about. Objective 2 is a lot of the translation on everything we've learned from the first SweetCap project, all of these sources of disease resistance and the sources of co-germination and so on, translating that and transferring that to elite materials, both public and private materials, that can hopefully become future parents for commercial hybrids. And that is the goal here for Objective 2. Objective 3 will study eating quality and nutritional properties of sweet corn. So here is where there is a component of it that's related to breeding, but there's also a component that's just the social science and the understanding of the survey of the consumer, to really try to understand knowledge gaps and how can we better educate consumers about the sweet corn health benefits and so forth. Objective 4 is a little bit more in the basic science, is testing a new strategy to engineer, and these are not transgenic plants, but to change specific genes And now that we know which gene, and now that we have some knowledge about which genes are important for some of the traits that are relevant to the different markets, we want to assess whether this two actually can be relevant in the context of breeding. This is being tested now in a variety of fruits and vegetables, and we definitely wanted to make sure that sweet corn is not left behind. Then the fifth objective, we have Paul Mitchell in the group. He is an economist, and then a lot of it will be an economic assessment of just micro and macro market pressures and price volatility, and really how sweet corn markets are changing over time. And he will be carrying a lot of these analyses. And I've reported some of that, the preliminary data in the past to different presentations in South Florida and Everglades. So these is just a brief summary. I hope to be able to explain over the next few years the different objectives and the different outcomes. And I welcome any suggestions that you may have. Please feel free to reach out. If you want to learn a little bit more about some of these points, please reach out and I'll be happy to expand on them. On the breeding side, it was a great year for us to strengthen the collaboration with some of the seed companies that in the past we had not collaborated as closely. So the UF breeding program continues to be an inbred development program. So we develop parents that can then become inbreds that can become parents in commercial hybrids if successful. So every year, we have now established agreements where the companies will actually send their elite inbreds to us, which enables us to actually generate a much larger number of crosses. So we create crosses between company inbreds, company parents, and UF material that's being developed every year. And thanks to the collaboration of many of growers and many of you who may be there in the audience, I really appreciate you allowing us to test some of these materials in your land. This is really valuable because it gives us the reality of the performance of these materials. So every year we're sending these hybrids to South Florida and evaluating them by selecting which ones are the best. So these are just 4 examples of hybrids that are coming back next year, and they will be tested for the 2nd year. And really good snap, really good ear package. Sizes are within the standard and pretty good flavor. So we're quite happy with these lines. We'll see how they go. In addition to these 4, we're actually going to be planting several hundred new combinations that were created this year between UF material and the different companies that we're now collaborating. So the hope is really to try to get all this research that's been and all these public funds that are being invested in the program here, and try to really translate that into genetic material that can reach the growers. So breeding takes a long time. I don't expect that to happen very soon, but I'm very pleased with how the program is evolving. So that's what I have for today. Please do reach out if you have any questions. This is my email, and I hope to see many of you, if not next week, next year for sure during the season. We'll be down there quite often. Thank you very much. Bye-bye.
So that was the update from Marcio, Dr. Resende. This is information, so if you need to reach out to him, please to do so. And let's keep moving. Let me see if I can get this on. Our next speaker is going to be Julien Beuzelin. He's the entomologist here at the station, and he's going to talk about more sweet corn insect ecology and management.
All right. Well, can you guys hear me okay? So let's talk about insects. We will talk about some of the work that we did with my master's student, Larissa, who is somewhere in the room over there. And I had planned to talk about those different topics this morning, But Anna informed me that I only had 20 minutes, so I had to make some very hard decisions. And so what we did, we prepared a handout with some of the things I will not talk about today. So we have it here, either as a standalone handout, or it's put in a binder with some additional information on seal fly identification that we've distributed at previous meetings. But if you want, so when we're done during lunch, please feel free. And those handouts, you don't have everything, but they talk about fall armyworm insecticides, cotton aphid insecticides, and then it's all about seal flies, identification, sampling, some of our insecticide resistance monitoring, and a lot of the lab work that we've done to try to identify new insecticides for seal flies. But today in this presentation, I would like to talk about these 2 points. So it's very focused. It's somewhat hardcore seal flies, but if you had to deal with them, you know that they can be pretty annoying, and we have to be pretty serious going after them. And so I want to talk about landscape ecology of corn seal fly. So why? Well, we try to spray our way out of seal flies, and so far it's working, but we have to be smarter. We apply insecticides and the adults come back into the fields for infesting. So we have this hypothesis that then the habitats surrounding the sweet corn fields, so that includes sugarcane and non-crop habitats, they are refuges for the seal flies. And so if we really want to improve seal fly management, we'd have to understand the role of those habitats and see how we can manipulate those habitats to try to prevent some issues. So we had to start somewhere. And so our first study was conducted to determine the relative abundance of seal fly adults in sweet corn fields, but in the adjacent habitats. So this was a study that was done exclusively on commercial farms in the area. We had 2 years, 2020, 2021, and then 2021, 2022. And a lot of that work, all that work would not have been possible without the help of crop consultants, especially Matt and Trey, identifying those sites and coordinating, very important. For each year, we had 3 seasons, fall, winter, and spring. And for each season, we selected 8 sites. At each site, then we had the 3 habitats: a sweet corn field, sugarcane field, and a non-crop habitat. And within a site, they were, well, not more than a mile apart. And then for each habitat, we put out 5 traps to sample seal flies. And we sample seal flies. We started standardizing with the corn around tassel push to early silking, and we monitored for 2 to 4 weeks. So we use those modular traps driving around. You might have seen them during the sweet corn season. And indeed, we did catch adult seal flies. I will primarily talk today about these 2 guys here, Euxesta stigmatias and Euxesta eluta. Chytopsis macilla, although adults can be pretty abundant in the corn fields, they are typically not as much of a problem. So these are the results for the fall here. So it's relatively data heavy, but I'm going to try to summarize it a little bit. So this is the seal fly abundance during the fall 2020 and then 2021, so the 2 years. And I'm reporting the number of adults per trap per day, so it's standardized. And so you can see that in both years, the lowest numbers were actually in the corn, thanks to the insecticides most likely, and the highest numbers were in those non-crop habitats. In 2020, actually, we had a statistical evidence for that pattern. If you take a closer look here, we still had a comparable pattern, but we did not have statistical evidence because this is the thing, there's a lot of variability in the data. They still give us a good idea if you look at those patterns and I report the species, Euxesta eluta in blue, Euxesta stigmatias in orange, and Chaetopsis macilla in gray, that we had the 3 species, the 2 years in the 3 habitats. But you can see that the relative proportions, they do vary. So what did we have during the winter? So here, what's interesting is those 2 winters we did that. We had a cold spell in either late January or early February. And each time one field was abandoned, one sweet corn field was abandoned. And actually, those sweet corn fields were not included in there because as soon as you start spraying, sailfly numbers go up. Incredible. So they are not out there. So these are the fields that were still managed. And you can see that actually in both years, we could not find differences among the habitats. We had sailflies everywhere, the 3 species in the 3 habitats in the dead of the winter. But now if we look a little bit more at species composition, you will see that the proportion of Euxesta stigmatia, so that's the orange, in the corn relative to the other 2 species was greater. So in other words, if you had to figure out the average color of each habitat, the corn was pretty orange, saying that stigmatias was really associated with the corn. That was the main species that was in there. If we look at the spraying here, so we had relatively consistent patterns between the 2 years with the lowest numbers in corn each time and the highest numbers in the non-crop habitats. And in sugarcane, we had intermediate numbers. Again, we had the 3 species in the 3 habitats. But if we look at the relative proportion of the species here, yes, we were doing a good job keeping the populations low. Here it was a little trickier, but the relative proportion of Euxesta stigmatias compared to the Euxesta eluta and Chaetopsis macilla was higher in the corn. So again, relatively speaking, a lot of orange in the corn, whereas in the sugarcane and the non-crop habitats, you see a lot of blue. Euxesta eluta was, relatively speaking, the most common. So this was the first time we're trying wide scale to look at sailflies thanks to the traps that we developed. And you see that, well, the 3 species, they do occur in all habitats from the fall to the spring. There's no question about that. The flies were generally less abundant in the sweet corn field, thankfully, because we're managing them. But you can see that we still had sailflies out there. And also there is extreme variability in the observations, as shown with some statistics that in spite of the very obvious patterns did not have any significance because we really have, in my opinion, some localized environmental conditions that play a role in infestations. And so this is why scouting is still critical in my opinion. So, well, the habitat surrounding sweet corn field, they are a refuge for the adults. So it would make sense that an improved management strategy would involve those habitats, some kind of manipulation. But what I was trying to show is that, especially in the winter and the spring, the proportion of Euxesta stigmatias relative to Euxesta eluta was greater in the sweet corn fields than in the other habitats. Euxesta stigmatias is somewhat less susceptible to insecticides, so that would make sense. But I think that there's also something to do with intrinsic behavior and preference of this species that contributes to those different distributions that we see. So how do we better understand this so that we can further understand the role of the habitats surrounding sweet corn? So for this part, actually, we stayed a little closer to home. We work here at the research station with spring 2021 and spring 2022. Why the spring? Because, well, this is the peak of the sweet corn season when the sailflies typically are the most challenging to control. And we did that here because actually we had a nice setup. We have a little bit more than 700 acres, but if you look at what we have, so here that's spring 2021 and spring 2022, and here we had mature sugarcane fields in the spring. Here we were later, the following year, we had newly harvested sugarcane fields. So we had those patches of landscape. We also had a young sugarcane that had been planted 5 months before. We had some corn at our station. So in 2021, if you can see here, here, and here, actually that was commercial sweet corn, so managed commercially. Whereas here, that's experimental sweet corn is still managed, but not as aggressively. And in 2022, we had some experimental corn, so it was managed, but not as aggressively. So you see, you have that mosaic of habitats. And to make things interesting, in 2022, we also had some flooded young racks. So what did we do? Well, we decided to blanket the station with a grid covering 400 acres, 154 points. And so those points, they were about 300 to 400 feet apart. So this is how the grid looked like. And at each grid point, we put a trap and we monitored the trap weekly for 4 weeks, counting the seal flies. And so then what we do is that the sum of the seal flies captured over the 4 weeks expressed as number of flies per trap per day. So again, it's standard, so we can compare that with those studies and everything. We mapped those population at the station level. And for those interpolation maps, we use inverse distance weighted technique in our GIS. So these are the kind of maps that we obtain at the research station when we monitor for seal flies. So here I'm showing Euxesta eluta per trap per day. So adults, males and females, 2021 and 2022. These colors, between the 2 years, we had differences in numbers. So these are relative comparisons within a year. So you can see in 2021, you had some intermediate concentration of Euxesta eluta here in that western part of the farm, and here in that southeastern quadrant, and some here. That specifically corresponded to the tallest sugarcane at the time at the station. So here it was mature sugarcane, whereas here it was plant cane, because the rest of the cane was actually harvested. In 2021, we had a hotspot here that was in the weeds all of a sudden, just like that. And here, mature sugarcane, not sure why. And that's what I was saying. You have sometimes very local conditions where you just have very hot spots. So what was happening in the corn with Euxesta eluta? Well, we had commercial sweet corn here, very low levels, maybe nothing, close to nothing, close to nothing. Here, also not much. Whenever the corn fields were treated with insecticides, we did not have any Euxesta eluta. However, where you had less intensive treatment, you had some Euxesta eluta. So you can start seeing what is happening at a farm level. So now this is Euxesta stigmatias, 2021. See, you have a hotspot here, have a hotspot here, you have a hotspot here, you have a hotspot here. Do you have a guess what was there? That was the corn. If you want to know where the corn is, just follow the stigmatias. They'll tell you. You have some stigmatias out there, but really this is where the hotspots are. Here, you want to guess where the corn was in 2022? Just follow the stigmatias. This is where they were. So this was really striking how we could see that distribution of Euxesta stigmatias, even in those fields that were commercially treated with, I would guess, pyrethroid applications at least 3 times a week, if not 4 times a week. Numbers were low, but they were there and they were staying in the corn. So Euxesta eluta was found in a diversity of habitats, including the cornfields, but it was not captured in the fields that were intensively treated with insecticides. We know it's susceptible to pyrethroids, so it's not really a management issue. And then at the landscape level, you probably have all habitats that can be sources and sinks. So have to consider everything. For Euxesta stigmatias, what was interesting, it was primarily associated with the cornfields, regardless of the insecticide treatments. It's less susceptible to insecticides, so that's somewhat understandable. But it really looks like that to some extent, corn is a preferred habitat that is acting as a sink if it's in the landscape, even if you have stigmatias around, my bet would be that they are coming. And if you don't do anything about them, then they'll infest your ears and you'll start rearing Euxesta stigmatias and creating a problem. And I'm saying that because sometimes I receive ears with a few maggots from commercial fields that have been perfectly managed, but a few maggots escape. So we put them in a cage, and here from this particular farm, we had 20 ears. And you see, you wait 3 weeks, and these are the seal flies that were in those ears. So just imagine if you had left them in the field, what would have happened. But also what was interesting, that was 100% stigmatias. So stigmatias is really the problem. We need to prevent it from getting into the cornfield. Need to put signs, stay away. And when it's in the field, then maybe you need to whack them. So how do we whack them? In the handout, we tried lots of things. Some with success. We still have a lot of work on that, and we think that a baits promoting ingestion will be helpful. But I was thinking about the fact that, well, if they really like the corn, what can we do for them to dislike the corn a little bit? So we had an insecticide evaluation this spring, and what we wanted to, how am I doing on time? Yeah, okay. We looked at kaolin clay and diatomaceous earth. And the way we looked at it, so we looked at the kaolin clay alone or with a parathorid, and we look at the diatomaceous earth alone or with a parathorid, and we also had the parathorid and with PBO and a non-treated check. And so if you're not familiar, kaolin clay and diatomaceous earth, they are minerals, powders that they just make the insects uncomfortable. And some of the fritids, the fruit flies, they are so much related to the seal flies. They are probably the closest pest to the seal flies. They've had some good experiences, so we thought that was really worth it. So we had this spring here, a typical small plot test, 4 rows wide, 34 feet long, 4 plots, 4 reps, 8 applications, once every other day for 2 weeks, starting at early silking. And for efficacy data, we looked at number of adults as estimated with sticky cards, and also collected 30 ears per plot to do ear injury and infestation. So this is how a spray looks like. So we tried to spray over the top. That was 20 gallons per acre, but we're still trying to be realistic. And I mean, we went for it. This is how the corn looked like. And we thought, well, maybe if the flies don't like it, we're onto something, then we could start slowing down. So how did it work? Well, that was a little disappointing. So, and that happens a lot with seal flies, unfortunately. So you see here, I'm reporting the number of adults per card per week. And these are the different treatments. And again, the different species here. You can see some patterns that would make sense with the addition of a parathorid would help. And the kaolin clay and diatomaceous earth alone, no benefit. With the parathorid, no benefit either. But even you see all those letters saying, hey, I mean, we had very weak evidence for efficacy. When looking at maggots per ear, if you're familiar with seal flies, you can see actually seal fly pressure was somewhat low. But now we had a little clearer patterns. But of course, our non-treated check, you see those letters A, B, C. That means that we're not different from anything else. Based on experience, you know, you can still learn something. Well, parathorids help decrease number of maggots. No evidence for PBO, at least like that, doing much. And the products we tested alone or with the parathorid, no benefit. So I think we need to continue working on landscape ecology and insecticides to try, and that's what Marcio was talking about, that behavior manipulation, trying to steer the seal flies away from the sweet corn if possible, or track them somewhere else so that we avoid having ears like that. And especially when the markets are slow and some sweet corn is staying in the field, there's really some problematic situations. Before I finish, a lot of that work, definitely more than just me, all the graduate students, technical assistants, The crop consultants, again, could not have done that work without them. Some sweet corn growers who are very helpful, chemical companies, and some grant money to do some of that work. So if you have questions, I'm around through lunch, and we have some handouts, and we'll be available for discussion if you're interested. Thanks.
Thank you, Julien. I know we are running a little behind, but if there is one question, we can try to answer. Anybody?
Yes.
One second.
When you were setting up at the weeds, were you looking at specific weeds to set up for, or?
That was smart randomization right there for you. So it was random, but representative and diverse at the same time, so that if a pattern emerged, we would possibly identify it. So we had a lot of elephant grass, but we had some areas where we had short grasses, other areas where it was more, some palm trees and other things. So it was relatively diverse. At some point, we thought that maybe there would be an effect of ragweed, but then randomly I selected some sites with some ragweed and they didn't work. Castor bean, they might not like it too much, but we would want to confirm. I don't know. One thing where we've had incredible hotspots is if you have an entrance of a field, something like that, and you have several ditches or random bodies of water staying there, chances are you'll have a hotspot there. It happened multiple times. So I don't know if there is some decaying matter there. But so, no, we are trying to be as diverse as possible so that we could potentially detect patterns.
Thank you.
Thank you, Julien. All right, so we are done with all the big scientific presentations. And and now we would like to thank our sponsors and then give them the opportunity to give some updates. So one of them is going to be BASF, and we also have a Cortiva update, and I believe a Syngenta. So
well, thank you, Anna. I appreciate it. How's everybody doing today? Just a brief touch on FFVA. Another thing that they have going is the IR4 program. So if you growers have any chemistries that you see out there that you could potentially use in your fields, there's a program through FFVA that's available that we could potentially get registrations or amendments to labels put through FFVA through any of the big manufacturers of the chemistries there. So just a shout out for Tori there. But just a real quick presentation on some of our fungicides and a little bit of herbicides. Just a brief one on Headline Amp. This has kind of been our staple for the past couple of years. 10 to 14 ounce rate. This is a 3 and 11 group. We do have a new 3 and 11 I'll touch about in just a second, but we want to use Headline Amp later on in the crop cycle. It's got a 7-day PHI on it. So this one we want to use a little bit later in the rotation, not as early as the one I'll talk about in just a second, but most of you have seen Headline Amp over the past couple of years. Revitec is our newest fungicide to enter the sweet corn market. It is a 3-way product with 3, 7, and 11 in this product. It does have a 21-day PHI, so we're trying to position this product a little bit earlier. It's got great control of all your major diseases that they talked about earlier today. Great on blights. The residual on this product is amazing. It lasts longer than anything out there on the market right now. And we've got a use rate of 8 ounces on sugarcane. It's a little bit higher. So at the 8 ounce rate, it's a little, more economical. The new product in this, the new active is the methyltrifluconazole, the group 3. And the molecule is really neat. It kind of articulates towards the disease. So it's a very broad spectrum and it kind of forms towards that pathogen. The next one is Veltima. Veltima is our specific corn product. We use a ton of this in the Midwest on tar spot. It's been a game changer for the tar spot pathogen up in the Midwest in Indiana and Iowa. Another watch out is the 21-day PHI. So we're trying to get this a little earlier also, but the residual on this product is incredible. You could almost skip a spray if you go early and get it at the right timing. It does have the plant health aspect in there with the pyraclostrobin, but it does contain the new group 3 methyltrifluconazole in there. All of these are SCs. They can be mixed with any of the pyrethroids or anything you're going out there with. No mixing issues. Nothing there. Here's just a beauty shot of, this is northern corn leaf blight, I believe, untreated in the middle 4 rows here and treated on the exterior. And the last one here is Armazon. This Calvin went over it this morning. This is a product we came to the market with a few years ago. Our suggestion is 0.75 ounces per acre. Does a great job with atrazine or metribuzin, anything you want to mix in there with it. It's also registered on sugarcane at a little higher rate, but make sure you get your COC or AMS in there with it. Very low use rate. This product does great on, really good on broadleafs, but also covers up your grasses as well. But want to go early post with this, and mixes great with most of your other herbicides. But that's it.
All right. So second sponsors presentation, just a quick update. Michelle is here. Michelle Hayes from Corteva.
Good morning, and I'm glad to see everyone again. Let me just start with a few things from Corteva. I would like to tell you first that we have taken a few products out of our portfolio. You knew about Laura's band being removed. We've got rid of that one. Another product that we sourced to another company is Lanade. So Lanade is no longer part of Corteva's portfolio. Now, got that part out of the way. Let me tell you about the new things that we're bringing into our portfolio. We have a new product that we're going to be promoting in this area called Ridgeback. It's a combination of sulfoxaflor plus bifenthrin, and it'll be great for aphids in sweet corn as well as silk fly, because it have a full rate of bifenthrin in it. More about our insecticides, because we are an insecticide company as far as I'm concerned. We have Intrepid, we have Transform, which is another aphid product that we'll be using, which is sulfoxaflor, which is a part of Ridgeback as well. Radiant, you know, for the ear protection, hits the standard in that market for leopard control. Intrepid is another one of those that would be there. And we have a combination product called Intrepid Edge. And Intrepid Edge, one of the things that's been holding it back a little bit has been the 10 gallons foliar for aerial application. We have been in the process, and I thought that was removed from the label, and we reduced it down to, I think, 2 or 5 gallons per acre. And I would check to make sure, but Intrepid Edge would be another one of those insecticides that would be a great fit for the lepidopter species in corn as well. As far as things that we're going to be working for, you may have read here recently that Corteva purchased a biological company called Stoller. So we're going to be looking at those nutritionals that company have in all these aspects of sweet corn, especially on the mineral soils to give you that nitrogen push, as well as we have one that will give you the phosphorus uptake and availability as well. And we'll be looking at it in other markets in this region too, such as cane, sugarcane. That is my update on our portfolio. I didn't want to be long-winded. I didn't put together slides, and this was impromptu. Michelle, did I miss anything that you need me to cover? All right, thank you.
Thank you. Like I said, short and sweet. And then we have one more sponsor update here. Let me see. This is Zach Langford and from Syngenta. And the other Syngenta folks couldn't make it today. And then the same thing for Valent, unfortunately, Craig Campbell, and then FMC, Ben Johnson. So, but we all would like to thank them for the lunch and for sponsoring this event.
Hello, everybody. My name is Zach Langford. I'm the retail sales rep for this geography. Just wanted to take a couple minutes just to provide a couple of updates. I don't have much in the way of product updates at this time. I did want to jump into just a reminder kind of to dovetail to Julian's presentation, just to remind the group that we do have a 2EE recommendation on Agrimec SC. I do have a printed stack that I will put out, or wherever you would like me to put the stack, that you can have, which you need at the time of application if you do utilize it. A couple of things to remind you guys on with this particular product is the intent of labeling this as a 2EE was for this to go in as a tank mix partner with another effective silk fly material. Making the intent of that was really, the purpose of that was because of the high intense pressure that you guys have on silk fly, and just rotating, have another mode of action in the tank with it for that. So you've got that. The other thing that I'm also going to give you a stack of out there is our crop care guide. Covers from seed treatments, herbicides, insecticides, fungicides, the whole portfolio that we currently have on sweet corn. So some of the things to note there are just some of the plant backs decaying. As you all know, that's something we all got to be mindful of, but I will have a stack of that out there. If y'all have any questions on that, please feel free to reach out to me. I am going to put my contact information up. Another piece I wanted to just update on is just personnel changes recently to our Florida district team. So if any of you guys remember Eric Rawls was our R&D field scientist out of Vero Beach, and Eric retired somewhat recently. He put 35 years in, plus I think he said 5 years of interning. So he put in a lot of time for us. To replace him is Ty Smith, who is here today. You can raise his hand. Ty is our new R&D field scientist. Ty is going to be focused on new chemistries and products for Syngenta, and he'll be running around this area and across the state. Another change that occurred on our team is Mr. John Taylor. Has 32 years with Syngenta, retired in the spring. We appreciate all his service to this market and our team. And replacing John is Kendra McCorkle. Kendra has almost 11 years with Syngenta so far. She's worked in at the R&D level in Vero Beach. She's helped run our soils lab, and most recently she was in a commercial support role as ag specialist for the state. And so welcome her to this new role. Her contact info is here, and she also is basically I-10 south the peninsula as an agronomist for us. So if y'all have any questions, anything that we can support you on, please feel free to reach out to us. Thank you.